Good afternoon, all of you. Myself, Dr. Anand Dede, Assistant Professor in Zoology. Uh, we have discussed in the base part first topic genetics, and here uh, we have discussed in the first topic the multiple alleles, and then our curriculum under the Shivaji University Kolhapur, we will discuss the topic multiple alleles. Objective of the topics are introduction of multiple alleles, definition of multiple alleles, characteristics of multiple alleles, examples of multiple alleles. Here, first of all, what is meant by multiple alleles? Multiple alleles is nothing but two or more different form of genes exists in the some species, or also two or more alternative form of genes are present in the species. Normally, individual has two alleles, and then during his study, the genetic Mendel also assumed only two types of alleles of one trait. There can be large number of possible allele forms in that some same population called as a multiple alleles. Here there are number of alleles are same to the different species, but Mendel's these are assumed only two alleles of one trait. Trait is nothing but the inherited character, as well as alleles means these are the two or more alternative form of gene. Gene is the nothing but the segment of DNA, and then multiple alleles. There are different types. Mainly, we have discussed the characteristics of the multiple alleles. Multiple alleles are located the same locus in the chromosome, and then here to alleles regulate the particular character, and the process of crossing over is not exhibited by the multiple alleles. Multiple alleles may show dominance, dominant and intermediate phenotypes. Phenotype means these are the external appearance of the organism, and then wild type of a series is usually dominate to the organism. The study of multiple alleles may be done in population, and these are the most of the characteristics of the multiple alleles. And then examples of multiple alleles, there are ABO blood group system, means there are different types of blood groups in human being. There are 4 different blood groups are seen. These are the ABO blood group system. There are A, B, AB and O. Therefore, a person blood group is determined by the pair of genes, is nothing but to the one pair inherited from each parent, because these are identify its own set of its molecules called as an antigen and located on the surface of the RBC, red blood cell. And then persons need a blood transfusion, donate the blood, and match the recipient blood, or complication will occur. There are 4 different blood groups are seen. Blood group O is called as a universal donor. Blood group AB is called as a universal acceptor. And then here multiple alleles are seen to the AB blood group system. Suppose RH factor, these are the major factor are seen to the multiple alleles. A person blood group type used are called as a rhesus monkey. These are the RH. Now say that RH. RH means these are the rhesus monkey, and then your RH type is determined by the different pair of genes to the ones that determine the ABO blood group system. And then RH positive and negative depending on the certain molecules are present, and a person who is RH negative will experience a severe immune system reaction, and if RH positive blood gets into their bloodstream. It depends upon the RH positive and negative. These are the major factor to determine the blood group AB positive, O positive, B positive as well as AB positive. Here AB blood group system, these are the controlled by the triallelic gene. There are 6 genotypes. Genotypes means these are the genetic constitution of the organism, and then alleles for antigen. Antigens are present on the RBC of the blood, and then two alleles are co-dominant and one another, both are dominant over the third. Here allele IA, capital IA, these are the produce antigen A, and allele IB, capital IB, produce antigen B, allele small I produce no antigen. These are the three triallelic genes are present, but there are 6 genotypes are formed to the AB blood group system. Here table show AB blood group system, there are genotypes to form the genotypes as well as phenotypes. Genotypes means these are the genetic constitution. Capital IA, these are the homozygous possible blood groups of their blood group A, capital I, A, capital IB, these are the blood group possible blood group AB, and heterozygous, capital IA, small I, these are the heterozygous capital A blood group, and similarly capital IB blood group B, and these are the heterozygous, capital I, capital B, small I, these are the blood group possible to the B, and small I, these are the blood group O. These are the blood groups types A and B have two possible genotypes, homozygous as well as heterozygous, and blood group AB and O only one genotype of each. And then table show these are the AB blood groups, and then antigens are present to the surface of RBCs are shown here. Antigen on RBC, antigen A, these are the blood surface of RBC, antigen B, surface of RBC, antigen A and B, and neither A or B, these are the antigen are present to the surface of RBC in blood, and antibody, these are present, antigen A and antibody in plasma, anti-B antibody are seen, antigen A, antibody A are present in the plasma, antigen A and B, there are antibodies are absent, and then neither A or B, there are both antibodies are present to the in the plasma. And then blood type, antigen react with the antibody, there are type of blood groups are possible to the cannot have B or AB blood and can have O or blood group, and then antigen present on the antibody A, antigen B is react with the antibody A, there are type of B blood group cannot have blood group A and AB blood group, and can have B blood group as well as O blood group. Antigen A and B, there are no antibody can be react with the, and there are type of blood group, possible blood group is the AB, can have any type of blood group, is the universal recipient, and neither A or B, there are antigens are absent, but both antibodies are present, there are type of O, can only possible blood group is the O, and then these are the universal acceptor. This is the antigen present on in the surface of the RBC. These are the RBC blood type, and then antigen A is present to the surface of RBC, antigen B are present to the surface of RBC, these are the react with the RBC, and then blood group A, blood group B, and antigen present to the RBC, AB antigen, and no antigens are formed to the surface of the RBC, they are to form to the blood group. And what is the significance of the blood group as well as application of the blood group? By know the blood groups of the patient, parents of blood group of their children can be predicted, because these are the after marriage and then formation of the new young ones, there are determined the how which type of blood groups are present in the children, male as well as female, and then also helps to the innocent people involved in the murder case and identify the real murder. These are the identify with the blood group, helps in the safe blood transfusion and used by the settle cases of disputed parentage in the mix of cases in the hospital. These are the no any person accident cases. These are the blood groups are possible to the accident case of the patient. These are the suffer to the loss of blood group, and there are important parts to the these are the live condition to the patient, and then support to the blood group. And another example of the blood group, a homozygous man having blood group A is married with heterozygous woman having blood group B, then what will be the possible blood group of their children? Here, blood group A and blood group B, these are the same to the example, blood group A and blood group B. So these are the homozygous man and woman having blood group A and B. And then principal means these are the multiple alleles, example based on the multiple alleles, and then to find out the possible blood group of their children. Their parents blood group A, blood group B, and then genotype capital I, capital A, capital I, capital A. These are the homozygous, and then cross with capital I, capital B, capital I. These are the heterozygous, and then result possible blood group of their children are A and B. And checkerboard show these are the male parent and female parent. After crossing, they are formed to the blood group A and B, and then genotypes and phenotypic ratio are 1:1. But these are the examples based on the multiple alleles. How can form to the blood group of their children after married, then form to the blood group A and blood group AB. And another example of the multiple alleles are coat color in rabbits. Coat color in rabbits means these are the skin colors. There are different types of skin color, colored agouti, chinchilla, Himalaya, albino, and these are the 5 kinds of skin color are seen to the coat color in rabbits. There are chinchilla variety is lighter in the color Himalaya, and Himalaya albino will have pink eyes and white coat color and black coat color tips like nose, tail and feet. But these are the coloring factor are seen to the rabbits. These are the example of the multiple alleles. Here table show the possible genotypes of their coat color in rabbits as given below. Phenotype, these are the full color brown. Alleles capital C. Possible genotypes capital C, capital C, capital C, small ch, capital C, ch, capital C, small c, dominant all over others. And then chinchilla capital C, small ch, these are the lowercase small c with ch, uppercase, upper scripts capital C, ch, capital C, ch, these are the homozygous, another capital C, ch, and then capital C, ch and C, these are the dominant over the Himalayan and albino. Himalayan, these are the small ch, here small ch, homozygous, another heterozygous, dominant over the albino. These are the characters are seen to the external morphology of the rabbit. Coloring factor, full color brown, these are the dominant over the all over the others. These are the capital indicates, alleles indicates capital letter capital C. Chinchilla indicates these are the capital C, small c and ch, and Himalaya ch, albino small c. These are the recessive to the all others.